I'm pleased to present our first two speakers, Dr. Mario Tenuta of the University of Manitoba and Dr. Dmytro Yevtushenko with the University of Lethbridge. These folks are here to give us an update on the Canadian Potato Early Dying Network, or CanPEDnet. And I'd like to thank BASF for sponsoring this session. BASF has received registration for Symegra insecticide in Canada for potatoes. The new active ingredient, brofenilide, in Symegra provides a unique solution for growers to use in the 2021 season. The addition of Symegra insecticide complements BASF's current potato seed care solution of Titan insecticide. Titan insecticide continues to provide proven performance and suppression of wireworms and control of early season pressures with Colorado potato beetle. For more information on the use of Symegra, connect with your BASF representative or join us on February 10th for a webinar about wireworm management strategies. Take it away, Dr. Tenuta.
All right, here I am. Okay, thank you, Stephanie. And it's such a great pleasure to be here with you today, across Canada and the world, and to talk to you about our effort called CanPEDupdate, the Canadian Potato Early Dying Network. I'm going to kick us off first, and then Dmytro is going to come later. So I'm at the University of Manitoba. I'm a Senior Industrial Research Chair in 4R Nutrient Management and professor at the university. And what you see in that picture there is a field that the left side had been fumigated and the right side had not been fumigated. You can see the right side is not doing as good. And this was in late August in Manitoba, not doing as good as the left side. That fumigation did do an improvement in the yield and the disease incidence. So that field actually on the right is succumbing to potato early dying. And that's what we're going to be talking about today. All right, CanPEDupdate. Why have we started this project across Canada? Well, several years back, Bernie Zebarth, who was with Canada and now retired in New Brunswick, and we had lots of discussions, and one big issue always seemed to be the low yields in Atlantic Canada for potatoes. And then in the West, in Alberta and Manitoba particularly, there's a need to increase delivery of processing potato in both provinces because there's been quite an expansion in processing capacity in those provinces. And I dare say the West is the potato capital now of Canada. And so there's limited area or in terms of fields. And there was an issue with older fields in potatoes that were not yielding that good, or they could be yielding better. And then of course, potato is a competitive business, producing potatoes. So, and processors are competitive, but the growers too, everybody's competitive. So we want to get more efficiency of production. So we came up with saying, what's a major constraint? Well, potato early dying was a major constraint that seemed to be ubiquitous right across Canada. The goal to reduce the severity of potato early dying is the major goal of this project, okay? It's a major yield limiting factor across Canada. We want to help growers deal with PED and increase that profitability of potato production. Now, our project started in late fall of 2019. So you can imagine with COVID, dealing with COVID all last year, our efforts were somewhat impaired, but I'm going to be able to give you here and Dmytro a good overview of what we've been able to accomplish and work that we've been able to do before the project started, and that is the basis for the project. Okay, so I got a lot of slides, so we're going to go really move through here fast. So who's involved? Who are the team members, researchers? I'm the PI. What you see on the left there is, top left is Bernie himself. Now, Bernie and I, we put in the proposal and stuff. Bernie really focused on the budget. I still don't understand the budget, a very complicated budget, as you can imagine, across Canada. And I was focusing on the work plan. Bernie's retired, and Dao Chen, to the right of Bernie there, is stepped in as the lead in Agriculture Canada. So Dao is the Ag Canada lead, and I'm the overall project lead. Then we have other researchers, Dimitro, University of Lethbridge, Katerina Jordan at the University of Guelph, Khalil Al-Bourabi at the New Brunswick government, and Vikram Bish with the Manitoba government. We also have grower groups involved, and particularly two grower group associates that are actually researchers in the project, Ryan Barrett with PI, and Dr. Mathuresh Singh with ACS Laboratories in New Brunswick. And then we have the cooperation and sponsorship by the Potato Growers of Alberta, Keystone Potato Producers Association, Manitoba, Ontario Potato Board, Potatoes New Brunswick, and PI Potatoes. And then there's industry collaborators as researchers and funders that shouldn't be in a smaller font. They're just as important as everybody else. Phytodata in Quebec with Hervé van der Heyden, Gaia Consulting with Darren and Debbie Gibson, Cavendish Farms, McCain, Simplot, NVAC, Bayer, and Syngenta. So a lot of players for a big issue for a big country. What is potato early dying? Well, it's early senescence and death of potato that robs yield. It's mainly caused by the fungus Verticillium dahliae and the nematode, root lesion nematode, Pratylanchus penetrans. Okay, those are the two major culprits. There's some other pathogens that can be involved. These are the dominant. Okay, it's a vascular wilt disease, which means it chokes the water supplying system of the plant. So under dry conditions, hot dry conditions, the plant can't provide enough water to some leaves, and you can see what ends up happening. There's an image on the top right of an area that succumbed early, and then in the bottom right you can see that leaves getting nice and little crispy there because couldn't carry enough water to those leaves, and they burnt up. Early death robs yield of large tubers, and these are the culprits. A little bit close up. Top left actually is a cross section of potato stem, and those black specks look like little pepper grains are the rusting structure of the fungus that will be added to soil with the residue of the potato. And as the potato, that residue decomposes, those little specks become released, and that is the fungus waiting in soil for another potato crop. Okay, and they can last for 10, 15 years in soil. Okay, now the bottom left images are the fungus growing in the laboratory. Okay, bottom left on a agar plate as a colony there, and in the right on our microscope, and those black clusters look like, I was going to say grapes, they kind of look like grapes. The black cluster, dark clusters are the microsclerotia, those pepper grains. Okay, now the thing on the right is a nematode. I love nematodes, they look beautiful. And you can see at the top near towards the head is this pin thing. That little pin-shaped object actually gets pushed and poked into roots, and then the nematode can follow through holes it makes and enters inside roots of potato, damaging the roots, causing, well, in effect, it stresses out the plant, and we think that it actually makes the plant more susceptible to the Verticillium dahliae. So the two together, almost like a double punch to the head of the potato plant, or to the feet, I guess the roots, and it really will succumb. But you can also have the disease just with each of them alone. Tends to that you need higher levels of each when they're not together. This is the life cycle of verticillium. I don't want to get into it too much here. The key thing is it starts in the soil with those rusting structures of microsclerotia. They enter the soil of potato in the springtime, well, after emergence, and then by the end of the season, they will have proliferated through the stem and leaves, plug them up, and the plant chokes itself without water, and then the plant dies, and the fungus produces more microsclerotia in the dying plant. Then the residues are added back to soil. So at the end of the year, we actually have a heck of a lot more verticillium in the soil than we started with because of this introduction of new inoculum. And then over years, a little bit of the microsclerotia die if we don't have more potato to allow them to build up levels again in soil. So over time, a portion will die. And if you really wanted to kill all the microsclerotia in the soil through old age, you probably have to go about 15 years without growing potato. Okay, well, who's going to do that? That's not, that is not, makes no economic sense. So we need to come in with some intervention. And what does it do? Here's some results from Manitoba in terms of looking at the size profile of tubers. And with more potato early dying disease incidence in the field, you can see that the 6 to 10 ounce and the greater than 11 ounce and, well, anything above 6 ounces starts to decrease when you have more PED disease incidence in the field. So in other words, the reduction is in the large tubers, not the small tubers. And actually with potato early dying, you can get actually more small tubers and a lot less larger tubers. This is problematic if we're getting paid based on our size profiles, right? Where we need to fill a nice box of French fries. You're not going to do that with the 3 to 4 ounce taters. Here's more example, in this case from a PhD student, Oscar Molina, with me, who's now with Agriculture and Agri-Food Canada as a research scientist. And you see on the top left, marketable yield as the verticillium increases. So verticillium increases. So this is actually verticillium in the plant that we scored. There's more of the verticillium in the plant, you have less marketable yield. It declines. And this, the other figures are showing that phosphorus is really important. The more phosphorus, the better we can have in terms of yield, and especially of the larger tubers. I'll say a little bit more about that. So we can also have effects of the nutrients in the soil, we'll talk more about, in terms of lessening potato early dying, but definitely for sure the presence of the verticillium is a big factor. So the project now is made up of several sub-projects, and the very first one is looking at the culprits, verticillium and root lesion nematode, and surveying them and relating them to potato early dying disease and yield, okay? And we're particularly also interested in looking at the species of verticillium and root lesion nematodes across Canada. So work that Oscar had done in Manitoba with me show that there are several different species of verticillium in soil. And you can see here that verticillium dallii, that's the culprit for potato early dying. It's prevalent in plants, in potato plants and in soil across commercial fields in Manitoba. Then the next common one was verticillium tricorpus, and then there's some other minor ones. Now this tricorpus is very interesting because it looks like dallii, and it can be problematic in interfering with the ability to count and score the levels of verticillium dallii in soil because it looks so similar. And I'll show you some information to back this up, but the first thing is just to show you that this tricorpus doesn't affect potato. Here's Oscar grew russet Burbank in the lab, and tricorpus doesn't do anything, but you can see the verticillium dallii on the right just knocks out the poor plant. And in the top right there is the fungus of the tricorpus. It very much looks like dallii. It produces those black sclerotia, or those pepper-like grains that rest in the soil. And so we can see those and we say, we have verticillium dallii. So it can really throw us off, and especially for a farmer, it can throw you off in terms of if you get a laboratory report and you have verticillium. You have to ask well, what species is it? Is it the dallii or the tricorpus, particularly in Manitoba? Now, in the rest of Canada, is that the case or not? Okay, now root lesion nematode is also very complicated. I was astounded when visiting Atlantic Canada and talking with colleagues that the high levels of root lesion nematode in New Brunswick and PEI, greater than 2,000 per kilogram, that alone can be a yield reducer. And Dr. Kempinski, who's retired now with Ag Canada, had shown that there's not only P. penetrans that causes PED, but there are some other species of Pradalanchus or root lesion nematode presence as well. They're not as damaging because they don't interact with verticillium to aggravate PED, but on their own, they could be problematic in very high numbers. So when you start getting lab reports, commercial lab reports back, it just says root lesion nematode, but we really don't know what species it is. It really makes a difference. Is it penetrans or not? Okay. And so on the prairies, when I started coming here, when I started working in the early part of the millennium, I was receiving reports from farmers across the prairies, Alberta and Manitoba, saying I got root lesion nematode. I'm going to do something about my soil. I'm particularly wanted to fumigate. Well, I would say, well, show me the lab report. And the lab report did say root lesion. And I would say to them, well, give me some soil. We looked at the soil, and then we said, I came to the conclusion it wasn't penetrans, but it was another nematode. And since then, I've had a PhD and 2 master's students who have surveyed soils right across the prairies. We've found that 30% of all commercial fields across the prairies have P. neglectus. So it's not penetrans. Neglectus, we found, does not reproduce on russet Burbank, in Manitoba in the field and in the laboratory, and also that its damage on potato in terms of yield reduction was, at least in Manitoba, we couldn't really make a link that it was reducing disease, or sorry, reducing yield. Now in some areas, colleague Dr. Ford in British Columbia, working with Ag Canada researchers in Lethbridge, found that in a rotation study that there was high levels of P. neglectus in some rotations, and potato yields were low in those rotations. So that and our work in Manitoba have come to the hypothesis that high levels of P. neglectus form on other crops, and then may damage potato in trying to feed on it. They're not successful in reproducing, but they will try to feed, and so in very high levels, okay? So, but anyway, it's very complicated, and that's the story for the West. Now for the East, there's more species out there, and so the complication is going to be really problematic when farmers are coming to me with 5,000 7,000 nematodes per kilogram of soil. We really need to know, is it the penetrans or not that is present, so we can make some recommendations on how to control potato early dying. So some work recently by Dao Chen and his team at in New Brunswick and PEI, and looking at surveying soils in those provinces, commercial fields, and looking at the wilt on the left side, so that's on the below axis, and yield. So you can see as there's more wilt, the yield goes down for New Brunswick, and the same thing for PI. Now the PI is scored as area of disease under the progress curve. In other words, let's think of it, the further to the right, the more disease in terms of you'd see really unhealthy plants in the field, okay, or dying early. And again, the yield goes down with that progress. So it's really backing up that more disease incidence in the field will have a reduction in yield. It's very important data to know to be able to tell growers, well, what is that reduction, you know? So here we're looking at it's more than 50% reduction going from the lowest to the highest, okay, in terms of wilt and disease. So in the severe issues, it's about 50–60% yield reduction. Okay. Now in Manitoba, I would be saying probably about 30% reduction would be probably the worst case scenario. It looks even worse in PI in New Brunswick. And here, and now from the same study with Dr. Chen, that part is CanPEDupdate, that the verticillium levels in soil by measuring their DNA. As that goes up, you can see that the area under the disease progress curve, in other words, the disease goes up on the left. And on the right, as there's more root lesion nematode, not a really clear increase in disease per se. So it gets a little bit murky with nematodes. And I don't want to, gets really complicated, but you just need a little bit of Pratylenchus penetrans to aggravate the verticillium to cause disease. So sometimes it's not about the numbers of root lesion or the Pratylenchus penetrans, it's their presence or not. And as long as you have the verticillium dallii present, just a little bit of nematode is enough to cause a double whammy on the plant, okay? Now in PEI with Ryan's team, looking at different rotation crops and looking at the root lesion nematode, this are RLN in blue, and in orange verticillium, you can see that grass, legume forages, and soybean and peas, legumes really support a lot of the root lesion nematode. That is to be expected. For verticillium, all the rotation crops really encourage its presence, okay, of the verticillium. So in terms of what when we're surveying across the New Brunswick, PEI, Quebec, Ontario, Manitoba, and we're keeping track of the rotation crops on the farms to interpret our data in terms of the levels of the pathogens and pests. And in PEI, comparing high and low level of verticillium dallii in soil, you can see that the low levels of the fungus in soil has higher yield, okay? And you know, that's a lot, that's 110, 90 bags more with the low inoculum. In terms of total yield, 80 bags for marketable yield, okay? And you can see that both the root lesion and the verticillium are higher in the lower yielding, okay? So again, backing up that the presence of these critters really hammers our productivity, okay? Now, in sub-project 2 looks at other soil-borne pathogens associated with PD complex, okay? And this is really led by Phytodata, which is a commercial laboratory in Quebec, and Dr. Hervé van der Heyden is the lead there. The pathogens are Colototricum, black dot, Spongosophora, Fusarium, Pythium, Streptomyces, Menthosporum, it goes on. And so these are all potential either related with causing PD or some other yield robbing or quality issue for potatoes, okay? And so this is pretty exciting. It's going to be a molecular-based determinations development that Hervé is doing. Okay, so here's an example of some results looking at different extraction kits for DNA, pulling DNA out of soil and then being able to then analyze that DNA for the pathogens. And here's an example with two of the pathogens, and saying that, you know, depending on the kit, you have different resolution and sensitivity in identifying the nematode of the pathogen, okay? So this is really important to be able to screen, to find the best kit that we can purchase for extraction in DNA and then use them to develop a reliable methodology. So I do like a kit that can be used for a whole suite of pathogens, okay? And so a lot of work is continuing with that with Hervé, and the idea is that to do a rapid throughput method for multiple pathogens of potato, and of course for the fungi causing potato early dying inclusive, okay? Related with that is a validation of real-time PCR protocol for the quantification of verticillium cross soils for Canada. And this is using PCR, polymerase chain reaction, laboratory protocol to quantify the verticillium DNA in soil. So Agriculture Certification Services with Dr. Singh in New Brunswick is the lead on this. And he's been working with my laboratory in this. They are taking the lead. We've been able to transfer some of our methodologies to them, and in terms of particularly in terms of the culturing of the verticillium and our initial PCR methods as well. And the soils from the survey in Project 1, so Project 1, are being used to calibrate this method to provide an accurate rapid test that we hope that is going to be available to you growers soon. So work that done in my lab, relating the level of verticillium in soil by the traditional method. So that's the top left, the traditional method. So on the bottom is verticillium inoculum in soil. My traditional method, which is to plate the soil and look for growth of colonies of the verticillium, related to a new method we developed for DNA analysis of verticillium. And you can see there's a relationship. As the traditional plate counting goes up, then the verticillium DNA levels go up. So always a lot better analysis when you add the microsporotia freshly to soil and then extract the DNA and do analysis. You can see the line is a lot tighter. Well, that's the way it is. When you use field soil, things get a little looser. But anyway, the method is decent to give us an indication of verticillium levels in soil. And here's data from multiple fields in Manitoba, and you can see the relationships, not bad actually, in terms of the traditional method on the bottom and the copy number or the DNA quantification on the vertical axis. So they go up together. So that is great. And we have 3 years of data there. And so that's good. So, you know, I have great confidence that we're going to be able to provide a method to you. But however, I wish I could say everything's easy. One of the reasons why we really wanted to do the DNA is because of this. So Oscar, through his PhD, found that in Manitoba about this tricorpus verticillium, and it looks like verticillium dallii. And here's 2 fields, for example, A and B, commercial fields in Manitoba. The top are the traditional plate counts. And you can see field B is much higher than field A. Okay. Now we go down to the DNA for verticillium dallii. Well, field A has verticillium dallii, but field B has verticillium tricorpus. So what we were counting as verticillium in the top panel by the traditional method was actually the tricorpus for B. Okay, that's what we were counting. There are 2 different species there. Really, B we're counting the tricorpus, and in A we're counting the dallii. Now we actually look at disease in the bottom panel. Well, which soil had more disease? The A had more disease. Okay, even though the counts were a lot higher for B, but it was the wrong species of verticillium. So this is what led us into saying, jeepers, we need a molecular means here to decipher these fungi, because you know, a farmer would say, wow, I got the humongous counts here of verticillium in B. I'm gonna go out there and nuke my field and kill that fungus somehow. Well, you may not have to, but field A was actually the one that deserved to have a control practices implemented, even though it had lower counts, because it was the actual fungus of concern. So I think I've convinced you the importance of this, of knowing the fungus. And so ACS is going further and developing very specific molecular means to identify the verticillium dallii. And here are examples of looking at relationships between the DNA quantification and the colony counts, and so for different provinces in Canada. So the key thing here is that the relationships seem to be a little bit different based on the province, and I suspect it's likely because of the differences in soils, and particularly maybe some texture of the soil type, and changing the PCR reactions and things like that. So part of this project is really trying to make sure that we can provide methodology here for rapid analysis of this fungus that's tailored for the different regions of Canada. We don't want to mess up by saying that something that works in New Brunswick works for Alberta when it may actually not because of the soils. So at sub-project 4, we're looking at reducing analysis variability through development of high throughput system for soil samples, for large soil samples. This is Hervé's doing this with phytodata out of Quebec. And so here's just some examples. Two fields same field in Manitoba, where Hervé grid sampled for DNA in soil and found that you can see the areas that are in red are hot spots for Verticillium dahlia. Left is before planting and after harvest, and you can see the levels go up a little bit up after harvest, which is normal. And here Hervé has done looking at different extraction kits, looking for the amount. That's the top 3 panels in red there in terms of the amount of DNA by 4 kits, and then the bottom is the quality. Now, unfortunately, the kits that provide the more quantity of DNA also provide the lower quality of DNA. So there's a bit of a trade-off there. So we need to come up with, Hervé's coming up with what's the best kit to have that compromise of enough DNA and good enough quality to give us really specific and accurate and quality results. And this is just backs that up in terms of looking at the different provinces in terms of quantity and quality. Again, we want to come up with kits that are good for across the country. Now, in terms of control products, in CanPED sub-project 5, through this is example looking at various nematicides and fungicides, and this is Dr. Al Mughrabi's work out of New Brunswick, and you can see that the stacking of these control products, Elatis, Vellum, and Serenade, giving the highest numerical yield, but definitely you can see there's an improvement. This was in 2020 in New Brunswick. And here looking at the root lesion nematode differences, not so many big differences. Some treatments really knocked out the nematode, but can't really say that was what's causing. Again, in terms of the disease reduction or yield improvement, again, it's because just having a little bit of Pradalenchus penetrans will aggravate verticillium and cause disease. You really have to almost get rid of all the penetrans. And here in Manitoba, similar type of approach and study, and you can see that we get an improvement in yield, slight improvement, in terms of cent weights, about 15 bags coming up with in terms of improvement, but nevertheless, this is for one field, another field, again, an improvement. Here, the improvement in terms of bags is better. It's over 40 bags. And so there's, what can be expected. It's not going to completely get rid of potato early dying, but these fields are not actually very bad in terms of yield. They're actually pretty decent yielding fields. And we got an improvement from them by using these products. Now, will it pay off to use all these products? We have to do a bit of an economic analysis on them. But this is excellent. I was actually really surprised to actually find an effect when the fields really weren't succumbing to potato early dying that much. Now, another approach that we're looking at in this project is looking at fumigation. So this is work that Oscar did with his PhD, and you can you see where the Vapam was applied, where the fumigant was applied? Now it's here where there's no weeds growing, okay? So, and the fumigants are really good at knocking out the weeds, knocking out the root lesion nematode, and can suppress and knock back a bit the verticillium. And in sub-project 5, we're looking at that and here's last fall in Manitoba, fumigating. We had a very early snowfall, and so we were fumigating. Actually, this is a good condition because we're bringing water in, and it helps the fumigation process by sealing the surface of the soil, so having that little snow there. So that was a good thing. And the fumigant goes on in the fall, the year before potato. And I'm pretty excited about this trial because we're stacking fumigation to strips of fumigation and then putting those fungi and fungicide and nematicides across that. So I'm pretty excited about this experiment where we're looking at the combination of these practices and seeing if we can get a yield boost. Now, in terms of another project, in terms of building soil health, this is one of the things that we identified as a problem with the reduction in yields across Canada for potato. And in Manitoba, Ontario, and New Brunswick, we have studies, trials looking at improving soil health and looking at the benefit to potato yield. So this is just a summary of fumigation trials with VAPAM in Manitoba. And the bottom, I want you to go all the way down to the bottom to the yield, 8% yield increase over all our trials that we've done to date in Manitoba. And that's pretty impressive in my opinion. Some fields we had a 20% increase in yield. And these are fields that actually, we didn't know what we were going into in terms of disease. And so I call this a random approach to fumigating fields in Manitoba, where you can expect on average an 8% boost. Some fields get a lot more. And so we knew that there's a benefit there. The reduction in verticillium in the soil is only 40%. Okay, and so it doesn't kill the verticillium; it just knocks it back a bit, enough to reduce the yield and improve yields. However, there's always a problem with everything almost done. This is Oscar's work, and here is VAPAM, and the knocks down the verticillium, so they went way down, stayed low, except after potato harvest, and we brought back the residues in soil, the verticillium levels jacked back up into the soil. It's because those plants were so full of verticillium following VAPAM. In other words, VAPAM is not a silver bullet. It's not going to control PED forever. It actually only controls the year of potato production. And so what we want to do in CanPEDupdate was to say, well, okay, let's get that boost, reduce yield, you reduce verticillium in the soil, and can we do some cultural practices to keep the verticillium inoculum levels low so we don't have to fumigate every year? And one of the things that always throws up would be organic matter. So this is some early work I did in Manitoba looking at potato early dying areas and fields, and really the most severe fields with potato early dying had low organic matter, and fields that were succumbing to potato early dying had low nitrogen, had high verticillium, and had some salinity issues, okay? And that's what you're seeing there, and in terms of the relationships to predict the disease. And so organic matter shows up time and time again as here in Manitoba. Again, the greater the soil organic matter, the less the disease in the field. There's 23 commercial fields in Manitoba that I did. And so building soil health, one of the critical things about building soil health is increase in organic matter. So Dao's team in New Brunswick looking at rotations in terms of a 2-year rotation and 3-year rotation with complexity of what the rotation crops are. And just, the work has really just begun. Some preliminary work looking at the results from the 2-year crop rotation study, and showing on the left the tuber yields, root lesion nematode in the center. And as you can see, there is a bit relationship that as the root lesion numbers go up, the yield goes down. And then looking at the DNA of verticillium dallii in soil, not so much of a relationship, but definitely a relationship with the root lesion in these soils. So in Project 6, we're also looking at in Manitoba a complex system of various treatments with fumigation to see if we can help increase yields with 1 year of fumigation and then do some soil building practices. And in this case, we're fumigating blocks of soil and then using compost cover crops, combinations of those, biofumigation with mustards, and seeing if after fumigation, by doing those and increasing the complexity of those combinations, can we build health so that the next time we go into potatoes, the 3 years after we have fumigated, that the the verticillium levels in soil are so low we don't have to fumigate again. So we just began this last fall, so looking forward to the results. And the last is the field evaluation and demonstration practices. These are demo projects, field projects that McCain and Cavendish are putting on in Alberta, Manitoba, and New Brunswick. I don't have results for that because they're demo projects. So you're asked your local Cavendish and McCain reps for what demo projects they may have, the field demos. Okay, lots of people to thank. I'm going to go to Dimitro here. And thank you very much.
So thank you very much, Mario. I will continue. Our potato research, my name is Dmitro, and I represent Potato Research Lab. I'm head of Potato Research Lab at the University of Lethbridge, and we represent the province of Alberta in the Canadian Potato Early Dying Network. So among Canada's provinces, Alberta is the third largest potato producer and the largest exporter of seed potato. And according to statistics of Canada, and I checked this yesterday, Alberta still boasts the highest average potato yield. Why is that? It's all because of soil, very unique climate with cold winters, warm summers, well-developed irrigation system in the southern region. All this makes Alberta one of the most productive places in Canada to grow potato. So as a result, our province is a home to the leading potato processors such as Lamb Weston, Cavendish Farms, McCain Foods, and others. Moreover, a couple of years ago, Cavendish Farms opened a brand new potato processing plant in Lethbridge, which will allow the company to more than double annual production capacity to meet growing demand. So a major limiting factor in profitable potato production is disease incidence, of course, both in fields and in storage, and among them, potato early dying complex. This disease can reduce the marketable yield from 30% to as high as 50%. The challenge is that currently there is no current potato cultivars registered in Canada that are resistant, genetically resistant to potato early dying disease. Therefore, management at this stage of PED relies completely on cultural methods to reduce the number of viable microflora in soil below that specific crop-specific thresholds in which disease can develop. These thresholds are not determined for Alberta, and actually they are not determined for Canada. That's why we have this Canada PED network. So the main causal agents of PED is the fungal field pathogen Verticillium dahlia, and to less extent Alboarthrum. This is, by the way, and the severity of PED also is amplified by combined infection, so the presence of root lesion nematode of Pratylenchus penetrans, as mentioned Mario. On these pictures, you see fields in Alberta during one of our past seasons with PED symptoms, and this is an example how we isolate pure culture of Verticillium and use it for identification at molecular level. So you just cut the stem. You can see that browning staining of vascular tissue, which indicates the presence of pathogens, presumably Verticillium. And then you place those tissues under sterile conditions on Petri dishes in special medium, and eventually you will be isolated to pathogens, and some of them will be Verticillium, which is shown here near the Petri dish. Both Verticillium species are soil-borne fungi that invade xylem tissue, disrupt water transport in plants, and cause foliar discoloration, necrosis, and actually, because it inhibits photosynthesis, eventually it leads to reduced yield. It affects water transport and photosynthesis. Now, the problem with Verticillium, as Mario mentioned, as everybody mentioned, is the thing that Verticillium was form so-called resting bodies, which called microsclerotia. And those resting bodies can stay in soil for many years, for decades, and even more, even in the absence of susceptible crops. And it complicates the control of PED greatly. Now, about thresholds, this is the most interesting question. As I mentioned, for Alberta, we don't know this, and for Canada, but there is some publications talking about other regions in North America. And those published economic thresholds, according to one publication, from 1 of 5 to 30 microsclerotia per gram of soil. But more recent work suggesting a more narrow range of 8 to 12. So root lesion nematodes is a highly pathogenic, Proteolenchus penandrus is a highly pathogenic nematodes of potato. And economic thresholds for the presence of these nematode in soil that causes diseases is about from 1,000 to 2,000 nematodes. On the right, you can see the life cycle, and on the left, you can see the photograph images taken actually in our lab, and you can see that those red arrows indicated lesions. So the synergy of root lesion nematodes and verticillium fungi and PED severity is thought to be to those wounds indicated by red arrows. So those wounds, the nematode cause in root tissues, entry points actually for verticillium fungi. And also the infection of roots are also thought to be increased by root exodus. So now we will go just directly to our lab, our recent work, which was done. We are working in the frame of Canada PED. So major objectives just to determine the presence of population level of the major causal agents of PED in Alberta for this province. Then analyze the relationship between pathogen level in soil, severity of disease symptoms, and potato yield to determine, so the most important question, what is the region-specific thresholds for disease development? And of course, we are interested to determine the efficacy of new commercial products to control PED under local environmental conditions. Also to see the impact of fumigation on soil microbiome and potato yield. I will briefly talk just for several minutes about research done in our lab by Colby Robertson. It's his master study. And he's right now with Cavendish Farms. So he did 3, and he evaluated the efficacy of new commercial products to control PED under local environmental conditions and impact of those fumigation on soil microbiome and potato yield. So we tested Velum Prime, which is active against nematodes, Elatus against verticillium, Fumigant Peak Plus, active ingredient chloropectin, and of course control, grower standard control for Alberta. And this is the concentration of fumigant used per acre. It was applied in fall and injection. In addition, we had 2 types of soil treatment with bed freshener on the top. You see this picture on the right, and power healer bottom during planting. And of course, we quantified yield through 10-foot digs. Next slide shows trial layout strips. Each strip contained, I think, 48 rows, and the length was from 400 to 800 meters. So as you can see, 8 fields. Each treatment was separated by controls, one field with no bed freshener, another with bed freshener. So as usually, according to the work plan, we go soil samples, shipping to lab, analyzing these samples for the presence of fungus, Proteolenchus, and very interesting, we also did metagenomic analysis in Quebec City for the effect, again, fumigation on microbial diversity. Here's the results from verticillium pathogen level. I will go very briefly, but the idea is that we can see late season rebound in the level of pathogen. This red line, it indicates the previous fall level. So we need to compare actually everything against a red line. And indeed, we see that with verticillium elatus, that it was clearly inhibiting the presence of verticillium in soil samples. Next. The same applies for nematodes, but in this case, of course, active ingredient was Velum Prime. The most important thing for us that tuber yield, how this tuber yield is associated with application of those controlled chemicals. And indeed, we observed increase in tuber yield, and it was statistically significant. So I mean, there's a visible difference. And as you can see in these pictures on the right, that fumigation was very efficient, both fumigation and vellum prime and Elatus allowed to produce, to harvest more potato compared with control strips. I should mention that we never used fumigation in Alberta. It's absolutely new for us, but we need to test this just for the future, although probably it's not the best thing that we would like to do in controlling PED. But anyway, we have to test this. And one of the very important questions was the effect fumigation has on microbial community. So those soil samples were also, as I mentioned, to analyze metagenomic analysis to done to see how much damage we did to microbial communities. And the idea is, the result is for those microbiome analysis that fumigation did not greatly reduce microbial species abundance or diversity, though there were changes to the community structure. So we did not observe negative effects of fumigation or application of fungicides to microbial diversity, although it's clear that microbial community structure was altered. We need to remember, though, that this was only one season experience of one field just for 1 year. Therefore, to fully evaluate the potential benefits or negative effect of fumigation, in our case, picloram, we need definitely additional trials. So to conclude that study, we can state that application of these chemicals, chloropictin fumigant, Elatus, vellum prime, reduced the level of both verticillium and nematodes in potato fields. However, this reduction was followed also a recovery of soil pathogen levels later in the growing season. It means something about something food for thought about how frequently you need to apply those chemicals. Now, these soil treatments significantly increased the yield of the potato crop, but were conditional to soil levels of target pathogens. Significantly, I mean statistically significant. Yes, there is a difference about control. Now, conditional to the soil levels, it means that it makes sense the difference was clearly visible only in those cases when the pathogens levels were high. If it's low, then it's practically none. Now, also we noted that using bed freshener was the best practice for the most efficient treatment with chloropictin. And finally, with microbial community, it looks like they were largely unaffected by soil treatments over the short period of time of the study, at least, with minimal or inconsistent difference from the control. Of course, we want to advance this research in the future, and our next task is identify these PED causal agents to species level and to the genus level. Then we are highly interested in molecular diagnostic, reliable and validated diagnostic, because as Mario mentioned, there is a difference between that and traditional methods to really show what's the present in your field. Also, we are interested to see the temporal aspect of PED disease development, about the early stages of plant colonization. And the ultimate goal, of course, of all us is to establish region-specific threshold for PED development in Alberta. So, and that, thank you very much, and we are ready for any questions.
Thank you, Dr. Yevtushenko, and thank you, Dr. Tenuta, for joining us today. We really appreciate having both of you here with us. We are just butting up against the time of the next presentation, but I'll take a few moments to hopefully have you answer a couple of questions. In your experience, Dr. Tenuta, has the P. penetrans and Verticillium dahliae interaction been primarily due to wounds on the roots, or increased root exudates, or a combination of both?
Neither.
Okay.
The literature indicates that it's actually a physiologic response, and initially we thought that it was the wounds allowing entry zones for the fungus, but it turns out that the plant itself, its immunity, if you call it, to the fungus decreases in the presence of the nematode in the root. So it weakens the physiologic response of the potato plant to defend against entry of and reproduction of the fungus inside the plant.
Okay.
And so the work done out of the University of Wisconsin has really shown elaborately this. And whoever asked a question or others, if you want to know more about that, please send me an email.
Great, thank you. We'll make sure that your contact information is available to our attendees today. Excellent. I'll just ask maybe one more here. Are there established thresholds for Verticillium dallii or root lesion nematodes where yields and long-term storage economic concerns are impacted?
Okay, so Dimitro was showing some thresholds that are used in the United States. So again, looking at the thresholds are designed or based on counting the number of colonies of Verticillium that are developed on an agar plate. And you don't need many colonies. So depending on 8, 12, 20, the thresholds are around there. So what ends up happening is that if you have the Pratylenchus penetrans nematode, a very specific species of root lesion nematode, you need the lower end of those counts of the Verticillium to have a disease. If you do not have the nematode, then you need higher counts. You're more at the 40, 50 counts to get disease and yield loss.
Okay.
However, as I was showing, our analysis for the counts can be thrown off if we have a particular species of Verticillium that is an imposter of dallii that looks like it, okay? And then those counts are useless. And that's where now we need to rely on molecular DNA analyses. And the step after coming up with a reliable molecular test of DNA for across Canada, is then to develop the yield response. Okay, so it's on our radar. Stay tuned for it.
Great, thank you. I'll ask just one final question here. Does potato early dying affect short season varieties, like a smaller table variety, at the same level as it would a longer season variety? Or is the effect just proportional to how long the variety is in the ground?
Excellent question. So cultivars of potato have different susceptibility to Verticillium. Okay, so that's one thing we have to understand. So our table and processing varieties, there will be a variance, and you could actually find a description for each cultivar, and there'll be a listing, and it'll tell you its susceptibility to Verticillium dallii and potato early dying. Okay, so there's one thing there. So you can look that up. In general, my experience is that the table varieties, which are harvested earlier than the processing, where we need to have a nice skin set and so forth for storage, the earlier you harvest your potatoes, the less you will see of a difference or a yield suppression. Okay, and so it really is late season. So let's say with russet Burbank, which is technically moderately susceptible. A lot of farmers say it's highly susceptible. That it's the tail end of the late tuber bulking stage, which is the susceptible time period for it. Okay, and so especially if that period coincides with high temperatures and low soil moisture, then you can really see a big effect there. So, but generally, yeah, earlier the harvest, particularly with the tables, then the less the the disease
Great.
and yield impact.
Okay, thank you very much.
Now, sorry, there was something
yeah.
mentioned there about storage.
Yes.
And so there can be issues with storage. I don't know enough about storage, but Rick Peters knows more about storage in Charlottetown. But there's always a concern too, especially with the secondary pathogens coming in cases of early 9 and having problems then for storage, for processing taters.
Okay, good stuff. Thank you. Thanks again, everyone, for your great questions. I apologize if we didn't get to them all today, but we will again do our best to get to your questions answered for you. And again, a great big thank you to Dr. Dmytro Yevtushenko and Dr. Mario Tenuta for joining us for today's event. Gentlemen, thank you so much. It was a pleasure hearing from both of you, and we really appreciate your time.
Thank you.
You're welcome.